So obviously, when we started our gene therapy journey, the first thing was like, okay, how are we going to get there? It's not easy to develop gene therapy programs. Even in the medicine today, there are very few gene therapy products that have been approved by the FDA or EMA. So the first thing was like, okay, who are the best scientists in the world that have advanced the understanding of the biology of hearing, and in particular, the understanding of the genetics of hearing? And the mapping of that exercise gave us the insight that Institut Pasteur is actually one of the top academic institutions driving the science of hearing. Obviously, we are French, so we built the bridge. And Institut Pasteur, just for the record, they are behind the identifications of the first genes, Otoferlin being one of them, that cause deafness. So then we partnered with them to make sure that their scientists could actually educate us, but we also built the capabilities internally within Sensorion. The second key question was like, okay, what's the best indication to demonstrate that gene therapy is a very good approach to restore hearing? And we picked Otoferlin deficiency because the babies are born bilaterally deaf. They are not suffering from any other type of syndrome. And from what we know from a biological standpoint is that the auditory capital that they have at birth is being preserved over time, which is very important for gene therapy. So that's the reason why we picked Otoferlin. So what's Otoferlin? Otoferlin is a protein that is being expressed in what we call the inner hair cells. And just also to maybe set a little bit of context, when you and I, we were born with only 3,500 inner hair cells. And every single one of them is tuned to a specific frequency. That's the reason why we love music. That's the reason why we can actually recognize the voices of our beloved ones. And this is where Otoferlin is being expressed. So that's the reason why we picked that indication. Babies born deaf, we know where the protein is being expressed. There is no degeneration of sensory hair cells over time. And the idea is that we can restore the expression of Otoferlin using gene therapy. At least that's what we demonstrated in the animal models. And not only we can restore the expression of Otoferlin, it is functional because in these animals, we've been successful in restoring hearing at the level of a normal mice.